Right, good evening. My name is Andrew Westby and I'm Director of the Natural Resources Institute and Deputy Pro-Vice Chancellor for Research and Enterprise in the University. And I'll be your host for this evening. So welcome to this lecture within the Faculty of Engineering and Sciences lecture series. And we're very fortunate tonight in having Dr. Francis Hawkes to talk to us about mosquitoes. So Frances actually did her PhD at the Natural Resources Institute, where she's working with Gay Gibson and Steve Torr, our colleagues, former colleague, on mosquitoes and how they fly and things like that. And that's led her to some really interesting discoveries, which has led them to doing some work on a mosquito trap, which is really exciting, but not actually the thing that she's going to talk about tonight, which is her work in Malaysia. That particular work was actually featured, or the work on the trap was featured on a documentary that was aired on the BBC back in 2015, was it, I believe. So Frances has the great honour, shall we say, if it's an honour or a privilege or something like that, of actually being the first student of the year back in 2012. And when she was a PhD student here, and she did her first degree here as well before that. So, she clearly has lots to offer, and it's a great honour for me that she's a part of the Natural Resources Institute. So I'm going to pass on to Frances, who's going to talk about human and ecosystem health, the case of Malaysia's mysterious malaria. Okay.
Hi everyone, thank you for coming this evening. I hope you can all hear me okay. If you can't, wave. I'm really excited to be talking to you about a project that I have been involved with since 2013, looking at mosquitoes particularly, but some much broader areas of science, including primatology, habitats, and human behaviour in Southeast Asia. This is what I'm here to talk to you today about, Plasmodium knowlesi. Plasmodiums are malaria parasites. There are a huge range of species of Plasmodium, and Plasmodium knowlesi is unique because it occurs in macaque monkeys. Now, we're probably most familiar with human malaria. We hear about this in the news quite often. Mosquitoes carry malaria parasites from person to person. But Plasmodium knowlesi is unique. It comes from macaque monkeys, where it's presumably been residing quite happily for many, many millennia. It's new to us in that we discovered it in the 1930s. Some poor rhesus macaque monkeys were taken from Singapore to the Calcutta Tropical Health Institute, where they were subject to research. Macaque research, primate research in the 1930s, as I'm sure you can imagine, it's probably not quite as enlightened as the type of research we might do today. But Plasmodium knowlesi was detected in macaque monkeys. This follows on from the work of Ronald Ross, who discovered malaria parasites inside mosquitoes in the 1890s. And he won the Nobel Prize for that discovery, because by understanding that the mosquito carries malaria from person to person, that those parasites can exist in the mosquito, we were able to develop new ways of controlling the disease itself. These parasites, these Plasmodium parasites, are the issue, not necessarily always the mosquito. Once Plasmodium knowlesi was discovered, it was kind of ignored. It was forgotten about. It wasn't really relevant from a human health point of view. It occurs in macaque monkeys. Well, we've got bigger problems to worry about in human malaria, which still killed, hundreds of thousands of people every year. So this is really a low priority for our investigations, but it was interesting and it was curious. I say it was forgotten about, but actually it had a sort of interesting sideline in science and medical history. Now, syphilis, coming from a completely different perspective, is a horrible disease, as I'm sure you are aware of. It's a bacteria, and there's very, very little you can do once you have syphilis. If you're in the 1930s, you contract syphilis, you will eventually develop neurosyphilis, and die. There is no treatment. However, an Austrian physician came up with a rather genius idea, which is that you can kill bacteria by heating them up to a certain temperature, and you can induce a certain temperature in a human patient by giving them malaria. The common symptom of malaria is a fever. So could you infect people with malaria to treat their syphilis? The answer is yes, you can. And again, another Nobel Prize was awarded for this discovery of pyrotherapy. The syphilis bacteria dies about 41 degrees, and you can infect your patients with Plasmodium knowlesi malaria and induce quite a severe fever which kills the bacteria. Now, it's worked in about 50% of cases. About 15% of the patients died of malaria, but they were going to die of syphilis anyway, so it was probably worth a bit of a punt. So this is thankfully something that we left behind when antibiotics were discovered, and you can treat syphilis quite easily now with a routine course of penicillin or antibiotics. So I'm sure everyone was very relieved to stop having transfusions of blood from infected monkeys and cycling various other diseases beside malaria between patients who were already very, very poorly. So Plasmodium knowlesi, apart from this rather curious kind of anecdotal, bizarre piece of medical history, was really forgotten about. Once antibiotics came along, there was no need to really pursue any further research into this very specific type of macaque malaria. If we fast forward to 1965 and the forests of peninsular Malaysia, we have a bit of an interesting situation emerge. There's a spy. Well, we think he's a spy. His initials are BW, and we know that he's a he. But beyond that, we don't know very much about him. All we know is that he is the only reported case of Plasmodium knowlesi infection in the last century. This is a human. He's become infected with a macaque malaria. He was probably doing things he shouldn't have been doing in the rainforests of Malaysia. We suspect he was actually a spy. He probably wasn't in a tuxedo. He was, out in the rainforest collecting radio signals for the communist regimes in the area. So that's why he's a bit of a shady character. But when his post was finished and he returned from Malaysia to the United States, he started to develop quite a serious fever. He was referred to the embryonic CDC, the American Center for Disease Control and Prevention, who analysed his blood and found that he was infected with malaria parasites, but specifically Plasmodium knowlesi parasites. So he's gone down in, again, the annals of medical history as BW, the patient who got monkey malaria, who was in the wrong place at the wrong time. And this was considered really just a fluke. It was bad luck that he got infected. He probably shouldn't have been, snooping around in the rainforest at night time and got bitten by a mosquito that had previously fed on an infected macaque. Now, he couldn't have got infected without the mosquito, which is my particular area of research interest. This is really what we think of when we think of mosquitoes sucking our blood. It's only the female mosquito that feeds on blood, and she uses that blood to develop her eggs. And she has this amazing set of mouthparts that probe into your skin and actually seek out your capillaries to feed on. But it's only so she can produce eggs for the next generation of mosquitoes. But it's this amazing set of mouthparts that makes mosquitoes the perfect creature for transmitting disease from person to person. We know that she sucks blood, that's the problem that we have. She has an evolved labrum, which is like a straw, to suck up our blood. But that's not all she does, and the problem arises when she injects you with her saliva. This creates a loop. It's a cycle. She's sucking up blood and injecting saliva, and it's that process that allows her to transmit disease from person to person. I hope you can see this video. It's a bit creepy. But this is filmed inside a mouse's ear. And what we're watching here is that proboscis entering into the mouse's ear. It's not like a needle as we imagine it to be, it's actually flexible and prehensile. So she can move around through the cells until she can find a nice plump capillary that she can puncture and suck your blood. But she's also injecting you with her saliva, which may contain parasites. So this is the slightly creepy side. I can see all of your faces. I wish you could see them. It's kind of alien-like. It is disgusting. But it's this incredible evolved ability to find and suck vertebrate blood that makes mosquitoes one of the most dangerous animals on the planet. And the malaria parasites exploit this behaviour to transmit themselves between people. It's a complicated cycle, but the malaria parasites have evolved exactly to be completely matched to the human and to the mosquito. They take different parts of their life cycle in the human and other parts of their life cycle in the mosquito. They depend on both. When the female has ingested your blood, it takes a little while for the mosquito to digest that blood, and likewise the parasites go through a change as well. They develop into small cysts on the outside of the mosquito gut, and then after about 14 days developing, they burst out of those cysts and travel up to the salivary glands, and it's at that point that that female mosquito is then infectious to the next person that she bites. And when we think about human malaria, which is always our main focus in research because it has such a huge burden on human health and misery, it's quite a simple cycle. It's that mosquito bite on an infected person, transmitted to another person several weeks later. But there are lots of different malaria parasites, Plasmodium parasites that infect birds, or as we were just discussing, macaque monkeys, rodents, and those systems are equally focused on that particular species of mosquito, the particular malaria parasite. The mosquito has specific host preferences. These mosquitoes may just feed on dogs, they may just feed on birds, and it's those combinations of behaviours and evolution that allow particular mosquitoes to spread malaria parasites to other species. For our poor spy messing around in the Malaysian jungle, he was very unfortunate in that he got bitten by a mosquito that had already fed once at least on an infected macaque monkey and then fed on him. So he really was in the wrong place at the wrong time. As I said, he was the only case last century of a confirmed case of Plasmodium knowlesi infection in humans. It really was considered solely a disease of macaques of no medical interest to the human medical community. But if we fast forward to Malaysian Borneo in 2004, our spy was in the jungles deep in peninsular Malaysia, and those original macaque monkeys from Singapore that were identified with Plasmodium knowlesi are based down here. If we come over to Malaysian Borneo, there are two Malaysian states in Borneo, Sarawak to the south and Sabah to the north. And there were some researchers in the early 2000s working out of Sarawak. They were investigating human malaria in Malaysia. Malaysia's aim is to be malaria-free by 2020 and has made very good progress in that regard. Bednet campaigns have been very, very effective, and the human malaria parasites, Plasmodium falciparum and Plasmodium malariae, are very much under control. Very few cases of malaria in Malaysia. However, they still are a few cases, and it's interesting to understand how these pockets of transmission still occur, what can we do to prevent them. And the researchers are, of course, keeping track of all of these cases, but they had some disturbing reports come in. In fact, they were very concerned because they were having reports of patients from a particular district hospital who were having very poor reaction to the treatment. So the treatments for human malaria weren't working in these cases. And what was more worrying is that their fever cycles were very rapid. Now, when you have malaria, one of the symptoms is a fever, which is how you can cure syphilis with it. But depending on the type of parasite that you're infected with, those fevers come in different cycles. In normal human malaria, falciparum malaria, those cycles are every 72 hours, and they correspond to the cycle of those malaria parasites bursting out of our red blood cells. So the clinical symptom of malaria, the fever, is a result of the parasite developing into a new stage and bursting into your bloodstream, making you very poorly. And each time you go through one of these fever cycles, you become more sick because there are more and more parasites circulating in your bloodstream. In these particular patients, this cycle wasn't every 72 hours, it was every 24 hours, which is very, very rapid. So they were becoming very, very sick very quickly. Their parasite load in their blood was increasing rapidly. And they were obviously suffering with frequent fevers as opposed to having a few days to recover and allowing the treatments to work. So the researchers, their interest and fears peaked because there are lots of other tropical diseases that can cause fever. They got in a boat, went up the Rajang River to Kapit district, which is a small district in Sarawak, and they visited the hospitals and they took blood slides from the patients. This is the typical way that you diagnose malaria in the field. You can take a blood sample, very simply, press it onto a slide, and then look underneath a microscope. Now, I'm not a trained microscopist and I'm not a clinician, but there are some people who have the eye for this, and this is their job, to all day stare down the microscope and look at malaria parasites. And depending on the species, you can actually identify the type of parasite that you have based on how it appears. I can't tell the difference between these. Good luck if you think you can. This is apparently Plasmodium knowlesi, infected in a red blood cell. Again, we've got Plasmodium falciparum, the common human malaria in Africa, and Plasmodium malariae, which is a common human malaria in Malaysia. So the team were a little bit concerned because it could well be that we have a parasite that looks like other parasites. This is really important to know which kind of malaria you're infected with because it determines what kind of treatment you have and what your prognosis may be. For example, falciparum malaria is very serious but tends to be treated quite quickly and you recover quite quickly. Malariae is slightly less serious, a form of malaria, but it's a bit sneaky. The parasites can get into your liver and hide in your liver, and it's this kind of malaria that can cause a relapse several years later once you think you're cured. So understanding the type of malaria that you're infected with is very, very important. So they were looking at these blood slides, really struggling to tell the difference between these species. They had no expectation that there would be Plasmodium knowlesi in these samples at all. There had been no reports since the 1960s with the case of BW. So they took the blood slides from the current patients, and they also had the foresight to take the beautifully preserved blood slides that had been kept by the district hospital for the last few years. And they returned to Sarawak and actually took them back to the Liverpool School of Tropical Medicine. And it was here that they used new genetic techniques to identify the DNA of the parasites infecting those red blood cells. And they found that it was not a match for the two types of malaria that you find in humans in Malaysia, falciparum or malariae. They found it belonged to Plasmodium knowlesi, which had been kept on record and had been analysed as an interesting case study because of its use in syphilis treatment. So they confirmed the first time ever the outbreak of what's called a zoonotic malaria. A zoonotic disease is one that comes from animals and can infect humans. This has huge implications for Malaysia because if you want to control malaria, if it's resident in macaque monkeys, maybe you need to control macaque monkeys. It's very serious because we didn't know how to treat it. We had to think of new ways of testing existing drugs, changing the dosage, changing the treatment regime to try and cure this new, quite serious malaria. And it was found that the local population seemed to have very low immunity to it. Normally with communities that are continuously exposed to malaria, there's quite high mortality in young children under the age of 5, And then once you survive, if you survive that stage, then malaria is like having a cold. You could have it every year, every 2 years, and it's like a bad case of flu. But we were finding that very young, very old people, everybody was reacting quite similarly and quite severely to this form of malaria. So the impact on the local population was quite huge, and in fact, by going through those historic blood slides, they were able to see that, in fact, this was the most common malaria in Malaysia over the last 5 years. This is just another map of Southeast Asia. Our hotspot down here in Sarawak in Kapit district. But looking broadly at these green and red loops around Southeast Asia, the red is the distribution of macaque monkeys, long-tailed macaques, the primary reservoir of this type of malaria. And the green dashed lines are the range of Leucospirus group mosquitoes. These are a group of mosquitoes which are associated with malaria transmission. As you can see, there's quite a lot of overlap between these two groups. And just pause for thought that there's probably about 800 million people living within the overlap of these two factors that are necessary for the transmission of Plasmodium knowlesi malaria. We could be looking at the beginning of something quite large in scale, with a huge potential to reach large population centres in Singapore, in other hotspot areas around Southeast Asia where there are large, dense populations of people. So this had all happened sort of around 2004. It seemed like a newly emerging malaria, and it seemed to be having quite a rapid sweep through the population and was having very serious human health consequences. As scientists, we like to tackle these kinds of problems with a hypothesis. We like to try and understand what is happening, can we do something about it? And for a newly emerging disease, like a zoonotic malaria, this is a very, very big question because there could be all kinds of factors influencing and affecting the dynamics between people, macaques, monkeys. But we have a hypothesis, and it was the hypothesis that we were trying to test in the project that I became involved with a few years ago. The last kind of player I would like to introduce in this story is this. Does anybody know what this is? No? No? This is a product that I would put good money on the fact that in the last 24 hours you've probably either used or consumed something that contains this product. This is the palm oil nut. So about 50% of store-bought packaged goods contain palm oil. It's an incredible product. It's very cheap to produce, it's a very efficient oil to produce, it's very stable at high temperatures, it's solid at room temperature so you can use it for baking, it's got natural preservative properties, so commercial companies are using this product very, very, very generously in lots and lots of different kinds of material products. It could be makeup, shampoo, conditioner, toothpaste, bread, cakes, biscuits. The chances are, if you've eaten a breakfast cereal this morning, it probably had palm oil in it somewhere. The biscuits we're going to eat afterwards, they probably have palm oil in too. It's a really common product now, and that has been a very rapid uptake of this product. This is the palm oil tree, the oil palm, and it grows about 10 degrees either side of the equator, so in the tropical region, which is also where you find lots of tropical diseases. These small fruits come in huge clusters. It's very economical to produce. In fact, this is the cheapest vegetable oil to produce. Each single nut is about 50% fat, so you only need half the land compared to the next best vegetable source of oil. Of course, you have to make way if you're going to do some agriculture. If you want to grow palm oil, you need to clear space. If it only grows 10 degrees above and below the equator, there's lots of land in that region. A lot of it is tropical rainforest. And there have been huge campaigns by national governments in Malaysia, Indonesia, Brazil, to clear rainforests to make way for palm oil production. And this is often done through quite brutal regimes of slash and burn. The project that I joined was with the London School of Hygiene and Tropical Medicine, and we were hoping to investigate what was driving the emergence of Plasmodium knowlesi in Southeast Asia and Malaysia in particular, and whether or not it had anything to do with the Malaysian policy of clearing primary and secondary rainforest for palm oil production. To do this, we needed quite an interdisciplinary team. We had primatologists working on macaque monkeys, people like myself working on the entomology, the aspect that deals with mosquito to transmit the disease. We looked at human behaviour, and we're also mapping habitat change and the scale and speed of that change. The project started in 2013. It was one of the first research projects to use drone technology to map habitat change. So this is actually an aerial shot of some primary rainforest here, some secondary rainforest up at the top, and a small area of plantation just here. This was taken in February 2014, right at the beginning of this part of the study. 3 months later, the exact same area was mapped again using our drone, and we found that more than half of that area had been cleared completely. It's not even partial, it's bare earth underneath. And this is the kind of scale of change that we were seeing even within the course of the project, which made designing our experiments quite a challenge, because you would go to work one day and the field site where you'd been working was suddenly on fire because the rainforest had been cut down and the remains set on fire. Our primatology team were working with macaque monkeys. So these are the original hosts of Plasmodium knowlesi malaria. This is a long-tailed macaque. Pig-tailed macaques can also harbour Plasmodium knowlesi. They don't seem to be affected in the same way that humans are, probably because they live with this infection quite commonly. Our primatology team tagged a number of individual macaque monkeys from different troops in Sabah and on a small island called Bangi to the very north of Sabah in Malaysia. And by tagging them with a GPS collar, they were able to track the movement of macaque monkeys over time and in response to these habitat changes that we were observing. So the primatology team found that actually the macaque monkeys have established home ranges, often deep inside the forest, and that they really stay within their territory unless they're disturbed. And you can see on the right here an area that's been cleared, ready for cultivation. Not actually sure this was necessarily for palm oil. There's lots of other agricultural clearing happening at the same time. These yellow dots represent the individual radio tracks from a group of macaques, and the blue dots here, the red dots here show where those macaques have been roosting. So you can see that over time, as land is changing, the habitat is changing, and the macaque behaviour is changing to follow. They still need to forage, they still need to eat, so they're changing where they are in the landscape. We focused on a particular area called Kampong Paradason, and you can see each of these crosses represents a household. A lot of the communities that we work with still live in traditional bamboo longhouses, so multiple families living in one long bamboo structure on stilts, and these are signified by these black crosses. And again, the yellow dots are tracks from individual macaque monkeys. And there has always been and continues to be some overlap between where people and macaques are found. Part of that is that humans tend to make a lot of rubbish, leave a lot of things laying around, and macaques are quite smart and they'll exploit that. So sometimes we can create an easy meal for macaques by leaving some fruit unattended or some rubbish untaken care of. But in general, this particular troop was fairly stable and tended to stay away from houses and roads because they actually really don't like confrontation with people. They tend to come off the worst for it, and they are often hit by cars when they're trying to cross roads. So they tend to stay in their territory. In this particular place where we're working, I showed you a picture of some rainforest on fire before. This was taken in this area. And a few months after that photo, we mapped again and found a large area of clearing and a corresponding change in the distribution of the macaque monkeys, who had never been previously to the north of this road, but were now being found from their GPS tracks a lot further north. They'd crossed this road. In doing so, they'd moved past all of these houses that we can see lining along the road. And then the next month, an even larger area had been cleared, concentrating and pushing those macaques right up into the northwest part of this image. So we're seeing this rapid environmental change. We're seeing immediate responses from macaque monkeys, and we're seeing that those are possibly having some interaction with human habitation at least, but not necessarily with people. They're certainly moving around in human environments in response to evading change. Moving on slightly now to the mosquito aspect of our research, which is where I spent most of my time working. Thinking back, it's important to understand the mosquito behaviour. Anopheles gambiae is the main malaria mosquito in Africa. We know a huge amount about its behaviour, what kind of animals it feeds on, what times of day, and it's this behaviour that's allowed us to exploit that behaviour in terms of control with bed nets, indoor spraying. We know this mosquito will rest inside. So we can use that understanding to control mosquitoes. In Plasmodium knowlesi, we had no idea what mosquito was transmitting this disease. We knew it would have to be feeding on macaques to become infected, and we knew it would have to be feeding on people to then transmit the disease to humans. It's quite difficult to capture mosquitoes with macaques, but this is what we set out to do. So we worked with Sabah Wildlife Department to handle and work with us in husbandry of a few macaque monkeys, and we used their odours to attract mosquitoes. We also used odours from humans to attract mosquitoes. Where we were working with macaque monkeys, we had to use a lot of personal protective equipment because it's not just Plasmodium knowlesi that can be transmitted via mosquitoes, but there are many, many diseases, including tuberculosis and hepatitis, that can be transmitted directly from non-human primates to humans. And of course, we're probably all very aware of the Ebola outbreak in West Africa over the last few years. This is another example of a disease, viruses that can spread from non-human primates, like gorillas, to humans. So we had to be very, very aware the whole time that we were working with macaque monkeys, not just for our safety, that they may transmit diseases to us, but that we could infect them with, a common cold. So we were required to wear a lot of personal protective equipment, which, as I'm sure you can imagine, tropical heat, really humid conditions, was very tough going on the field team and myself. But by doing this experiment and working with humans and macaques and seeing which mosquitoes were attracted to each, we were able to identify Anopheles balabacensis. This is the only chart in the whole presentation, so I hope you appreciate it. This is a human-baited trap on the left and a monkey-baited trap on the right. And Anopheles balabacensis was the species of mosquito that we found in both. We then took these mosquitoes and we dissected their salivary glands. If you can imagine dissecting something very, very, very small and then make that even smaller, this is what it's like to dissect a mosquito. But we can dissect their salivary glands, and this is where we can see the parasites that transmit malaria ready to be injected into the next animal that the mosquito feeds on. So Anopheles balabacensis is our prime culprit for transmitting Plasmodium knowlesi from macaques to humans. We found it both attracted to humans and macaques, and we found it infected with the parasites. We also wanted to look at human behaviour, because this is probably the most complicated part of any system, is knowing what people will do. Animals can be quite predictable in some ways, but humans have lots of different rationales and reasons for doing things. So as well as GPS tracking and macaque monkeys, we did the same with some volunteers from our local community. On this figure here, you can see these yellow dots, they correspond to humans. And the blue dots correspond to macaque monkeys. And our green dots show where we were doing mosquito sampling. So we're trying to work in areas where we can see habitat change, where we can map the scale and the rapidity of that habitat change, and see what happens over time. And in fact, you can already see that these yellow traces from people are quite overlapped with the movement of the macaques, these blue dots here. And in fact, in this case, there's a small river running just through here, down into this clearing here, and it was actually discovered that this human behaviour of going to wash in the river, which is quite a common practice, was a real risk factor in people picking up knowlesi malaria, because they were going in the evening when the mosquitoes were biting, and they were overlapping in their land use with the macaque monkeys. So we were trying to take a very holistic and comprehensive look at the factors that are affecting disease transmission. So our project findings, well, macaque monkeys respond very, very quickly to habitat change. Although their home ranges are preferably deep in the forest and they prefer to be undisturbed, they will quite readily move through more settled environments. They'll come to the edge of villages and habitations to evade dramatic landscape changes. I can't stress really enough that the type of land clearing that happens to make way for agriculture is really rapid and violent. There are some fairly horrific footage of animals like orangutans being caught in slash and burn campaigns, and they really just can't move out of the way quick enough. But macaques, they will respond and they will move. But as their habitat shrinks, so too does the area that they can forage from, and this could well be concentrating macaque numbers in particular areas. We identified the key culprit, Anopheles balabacensis, the mosquito that's transmitting Plasmodium knowlesi from macaques to mosquitoes. And my colleagues in Malaysia have actually been able to colonise this mosquito. They're quite fussy, they really like very particular conditions, but by keeping them in the laboratory, we're able to understand their life cycle better. We can understand how quickly they need to feed to reproduce, which in turn has an impact on how often they'd be seeking a blood meal from a human or a macaque. And we're developing this study at the moment. We found that there are some human behaviours that could well be increasing the likelihood that people are exposed to Plasmodium knowlesi malaria. Traditional activities, washing, bathing, cooking, cleaning down by a river in the evening is a high-risk activity. But we found that people indoors were probably at lower risk than people outdoors. So even though traditional homes are very open, the bamboo structure is quite porous to the environment, mosquitoes can enter, we think that Anopheles balabacensis really doesn't do that. They won't come inside to feed. And we found that the scale and rapidity of habitat change was really quite massive. And large areas of our study sites that started as often secondary forest, but some primary forest, have now been completely converted into specifically palm oil cultivation, but other areas of agriculture as well. It's very hard to prove conclusively that all of these things are linked. There is a good chance that the scale of change and the speed of change has driven some of these processes, but we always have a suspicion that maybe something had happened before to instigate this emergence of the disease. And actually, there is an argument that we couldn't really have detected it before because we lacked the DNA tools to understand the diversity of Plasmodium species that existed in Malaysia. And if you don't know how to look for something, you won't see it. So it's possible that historically things had been misdiagnosed as other kinds of Plasmodium species. So what? That was kind of a bummer, right? It's huge landscape change. We're setting fire to tropical rainforests. We're destroying habitat for endangered species. We're causing horrendous diseases for ourselves in the process. It's not entirely doom and gloom, but there are some important questions that we need to think about. Palm oil is a product that I've used today, I know I have, I'm sure many of you have as well. It's a very efficient product to produce, it's very cheap, and there is an argument that there is a space for palm oil that recognises its environmental and social impact, and we should think about whether or not we're prepared to pay for that. Are there things that we can do to think about the complete effects of something? We, especially in commercial settings, we can become very focused on the output, which is a product and a cost of the product. But increasingly, people want social responsibility in the companies that they buy products from. Can we think about environmental, social, and health aspects as we do so? And what can we do to empower local communities? Because they are ultimately the ones that bear the brunt of these huge global economic changes. How can we empower them to take an ownership and responsibility and drive change that they wish to see in their own communities? There has been some work towards this. There's been a roundtable on sustainable palm oil production, so you may find a small logo on products now, similar to the Fair Trade Standard, that seeks to address some of these concerns, particularly with palm oil, because it's such a hugely important crop. Products should try and meet global food demand. There is a demand for this product, that's not going to go away. But they should be looking to support poverty reduction, safeguard the communities that are involved in these huge landscape changes and are often employed at the same time through the same systems. We want to support food prices that are affordable for everybody, whilst also protecting the environment and wildlife. All of these things can be quite hard to achieve, but there are companies working towards them, looking for accountability in the suppliers of palm oil. One thing that's not mentioned on here is health. Now, this is obviously the focus of my talk, how this huge business of palm oil production could well have induced the development of a new zoonotic disease in humans, but it's not currently addressed by the Roundtable on Sustainable Palm Oil, and perhaps it should be. Part of the difficulty with that, as I said, is proving directly the links between these processes. Nonetheless, I think it's important to recognise that as we change things in our environment, of course there are often unforeseen changes that follow on that we can't predict and are very difficult to show cause and effect. But we should be aware of these things. I'd like to say, before I finish, thank you to the teams in Malaysia who have helped with this project, particularly at the University of Malaysia in Sabah, and the team from the London School who conceived the original project. But I'd also like to extend thanks to the communities in Kampong Paradasan that we worked with, because they are the ones who are experiencing these changes, and they are the ones who are at risk of Plasmodium knowlesi malaria. There is some recent effort to get the World Health Organization to recognise Plasmodium knowlesi as the 5th human malaria parasite, although it is zoonotic in origin from macaque monkeys. And we're hoping that this should lead to new funding available to try and find solutions to this problem. Now that we've identified it, now we're getting a better handle of the drivers and understanding some of the biological systems involved, we need to think of solutions, creative ways of addressing some of these problems. So thank you very much for listening. I hope you enjoyed.